Welcome everyone to the LRIG scientific webinar. My name is Lorena Kallal and I'm at GlaxoSmithKline in Collegeville, Pennsylvania. I am your WebEx host today. Just a couple of notes about the WebEx. We are recording the session for future viewing. Secondly, participants are muted. So for the questions at the end, we will be using the chat function. You will click on chat and then in the drop-down, select to all panelists. And one of the panelists will read the questions to the speakers. With that, I would like to hand things over to the chair of our LRIG Philadelphia chapter. Over to you, Rodney.
Thank you, Lorena. Good afternoon. Welcome to the 6th virtual scientific meeting presented by LRIG Philadelphia chapter. You can find out about our events by going to lrig.org. This virtual scientific meeting is an opportunity for scientists to share their research and innovations with the global LRIG community. All our events are free to attend and open to all. If you are interested in organizing or presenting at a future meeting, please just email your proposal to talks.phl@lrig.org. I want to offer a special thanks to Lorena, who you just met, and who will also be our speaker, one of our speakers today, Ron Ferguson, and Sharon Matamoros, who will very shortly introduce our speakers. These three people have been instrumental in making our virtual scientific meetings possible. So I'd like to thank them. On Sharon, you can now introduce the speakers. Thank you.
Thank you, Rodney. This is a great presentation today. And welcome everybody to our integrated presentation about high-throughput cellular Tellerman shift assays, approaches and applications. First, I'm going to introduce Lorena. Dr. Lorena Kallal is a scientist and manager at GlaxoSmithKline in Collegeville, Pennsylvania. As part of the screening, profiling, and mechanistic biology department, her team supports programs across the GSK portfolio with cellular assays and experimentation. Lorena has expertise in high-throughput screening, phenotypic assays, and drug discovery strategy. So Lorena is going to be our second speaker. So now I'm going to introduce our first speaker. So you can please move the slide. Thank you. So our first speaker is going to be Mark Henderson. Dr. Mark Henderson is a biology group leader in the early translation branch at the National Center for Advancing Translational Sciences. His team works on collaborative projects aiming to identify small molecules for disease-relevant targets, focusing on cellular processes that are dysregulated across multiple disorders. He has experience in the design, implementation, and development of high-throughput screening assays. So first, we're going to pay attention to Mark Henderson presentation, and after that, Lorena will continue the presentation. Thank you, and please, Mark, start your presentation.
All right, thank you, Sharon, for that introduction, and thank you for the invitation to present today and share some of the work that we've been doing on high-throughput CETSA or cellular thermal shift assays. And I'm particularly happy to be sharing this presentation with Lorena because I think our groups have have followed similar journeys over the last couple of years in terms of learning about the strengths and the limitations of cellular thermal shift assays, particularly high-throughput versions. And so I'm looking forward to sharing some of that today. Okay, so I'll start with this schematic that places CETSA kind of within a typical small molecule discovery program. And so Many target-based screening efforts start with a biochemical assay, a simplified system where you're looking for small molecules that alter the function of a target of interest. In many cases, it's a purified protein, and then you can either look and ask whether your small molecule engages your target using some sort of biophysical assay, or you can look at a biochemical response, for instance, an inhibition of enzymatic activity. When transitioning from the biochemical setting into a cellular system, it's often a high hurdle for a program in terms of maintaining activity of compounds into the cell. And there's a number of reasons that compounds may lose activity as you go to a cellular environment. The first is that you have to cross a plasma membrane, at least for intracellular targets, and in some cases you have to cross multiple membranes. Once you get inside the cell, there's a much more complex environment, and now the molecule can start to bind off-target proteins. And ultimately, if your molecule does hit your target, there's a couple of ways that you can assess this activity, and typically it's either with some sort of phenotypic readout, like an inhibition of growth, or maybe a functional readout, like a change in metabolite concentration. But in some cases, these types of readouts can lead you astray in that they can be altered by off-target activity as well. And so having an assay that kind of sits in the middle here that allows you to look closer and determine whether your molecule directly engages your target is an important step in this process. And this is where target engagement assays come in, and CETSA in particular has become increasingly popular over the last several years to look at this binding event within a cellular environment. So I'll introduce really briefly this target, lactate dehydrogenase, because I'll use this as an example throughout the presentation for different high throughput CETSA assays that we've run. And so this target here is of interest to a number of NCATS scientists for its role in cancer and a number of other human diseases. And so the idea here is that in cancer condition, you have increased glucose uptake, and this glucose is shunted over and converted from pyruvate to lactate. And so by inhibiting this enzyme, you can have a beneficial effect and potentially kill cancer cells. And so in terms of a cell-based assay, you could measure lactate levels in the cell as a proxy for inhibiting this lactate dehydrogenase. But of course, metabolism is extremely complex and interconnected. And so there's multiple different mechanisms by which you could change lactate levels in the cell. And so as with all of these assays moving into a cell-based environment, it's important to confirm that your molecule is acting through an on-target mechanism to drive a phenotype, as opposed to an overlapping phenotype that's driven through an unintended target. And so this is where CETSA is a nice approach to be able to look directly at the target of interest. And so the cellular thermal shift assay, as I mentioned, target engagement assay, and the premise of the assay is that within a cellular environment, protein will unfold and aggregate at a given temperature, depicted by this dark black line here. And a small molecule binding event may alter that aggregation temperature. And so in this case, the small molecule binding is stabilizing the protein so that when it's heated, it takes longer for it to aggregate and unfold and aggregate. And so traditionally, the CETSA assay, the readout is to use an immunoblot strategy, a Western blot. And so after heating, the lysate is centrifuged at high speed, and the insoluble fraction is removed from the lysate, and then the soluble fraction is run out on an SDS page gel. And in this example for LDHA, what you could see is that at 65 degrees C, the protein has aggregated and disappeared, but in the presence of the small molecule inhibitor, it's stabilized and maintained soluble up to above 70 degrees C. So the CETSA assay requires good antibodies that are specific, and of course there's throughput limitations when using Western blot as a readout. And I like to give the example of if you wanted to do a screen of the approved drugs collection, which is about 2,000 compounds, and you wanted to test just one concentration for binding to one target by CETSA, it would take somewhere between 100 and 200 Western blots. So clearly this isn't a high throughput assay and would take too much effort in order to do this type of a study. And so thinking about ways to increase the throughput and move these assays into either 384 or 1536 micro-well plates is a goal in order to be able to have more functional CETSA assays. And so a number of groups have done this over the last several years. In some situations, the endogenous protein is measured using a pair of antibodies and either alpha screen technology or HTRF. And this is advantageous because you're measuring the endogenous protein, and it's also a homogeneous assay. So there's no centrifugation and separation step required. You can heat the samples and drop in the antibodies and directly measure the amount of soluble protein that remains. The other approach is to fuse reporter proteins to your target of interest, either full-length reporter proteins or the split reporter systems. And so both myself and Lorena are going to talk about split reporter approaches, in which a small portion of the reporter is attached to your target of interest, and then the remaining portion of that reporter is added at the detection stage. So after heating, the soluble protein can be quantified by adding in the additional part of the reporter protein. And these are advantageous in that the small nature of the split reporter system is less likely to affect the protein's function, and also less likely to cause issues in terms of the aggregation profile. So you might imagine that if your full-length reporter protein aggregated at a lower temperature than your target protein, that may actually confound the interpretation and prevent you from performing a CETSA experiment. So the approach that we've focused on primarily is the split loop CETSA approach. And the idea here is that we've used a 15 amino acid peptide tag. And this peptide tag contains the high-bit, high-affinity peptide sequence that was developed by Promega that can bind to a large fragment of nanoloop variant. And so the idea of the assay is that when the protein unfolds and aggregates, it's no longer available to bind to this large fragment. And you can measure the remaining soluble protein by adding the two components together with a furimazine substrate and measuring light emission. Importantly for throughput, this assay doesn't require any centrifugation step. So this is not LDHA, but another target with this peptide appended to either the N terminus in red or the C terminus in blue. And there's lots of soluble protein. And then if we heat to a high temperature where the protein is expected to be aggregated, and then we spin that sample, we lose soluble protein and the signal drops down almost to baseline. But what is useful for throughput is that you get the same response when you don't centrifuge. So the way that we envision this is that the protein, when it unfolds and aggregates, it goes into these big, large aggregates that are no longer accessible for the large fragment of the enzyme and the furimazine. And so you can do it as a homogeneous assay. And we've seen reliable compound-induced thermal stabilization for targets that we work on that we had traditional sets of data. So this is LDHA, similar to the Western I showed, where in the presence of vehicle, it melts at a lower temperature than in the presence of a small molecule inhibitor. This is an overview of the entire assay procedure, in which you transfect in your target of interest that has this tag on it, wait for a day or two and allow the transgene to express, lift those cells, and replate them out into plates, add compounds, heat, either using PCR plates or microwell plates, lyse the cells, and then add the large fragment in furimazine and measure light emission as a way to measure the soluble protein that remains in the sample. So one of the first challenges that we faced when developing this high throughput CETSA and moving from PCR plates over to microwell plates was getting a reproducible heat, reproducible melting and aggregation across the entire plate. And so this is some data from early on where we tested heating plates in an oven by convection. You can see there's quite a lot of variability, and this is as the wells heat kind of from the outside to the inside of the plate. And so we moved to a heat block approach where we machined an aluminum block here and then placed the plate on top of it and put some top pressure on it, and we get much more reproducible heating across the plate. And this depicts a typical optimization experiment that would happen early on for a target for these high throughput CETSA experiments, where we dispense compound to every other column of the plate. either vehicle control, and those are these low luminescence samples where the protein is aggregated, and this is a positive control sample. And this allows us to dial in on the optimal conditions for both temperature and time of heating for the target. And this is example data for a couple of targets where you could see as you increase the heating duration in these lighter gray bars, the protein starts to melt, and then you can assess assay performance plus minus your small molecule control. To this point, we haven't figured out a magical formula for going from a PCR plate to a microwell plate in terms of time or temperature, and we've had to do the work and figure it out empirically for each target. At least to this point, we haven't observed anything that would allow us to predict the appropriate temperature or time. This is just a picture of a second generation heat block where the block is controlled by a controller, and there's a thermal probe that's into the center of the block. And then, as I mentioned, we found it's really important to put top pressure on these plates. And so this is a plate that would sit down on top of your plate, and you could clamp it down with screw clamps. Because what we found, if you don't put that top pressure, the plate starts to bow, and you get artifacts across the plate from heating. So this is an example of what a plate would look like for a lead optimization type campaign. And this is a plate that isn't heated, and you can see a nice strong signal across the plate. And then if we heat the plate in the vehicle control treated space, the samples disappear as the protein aggregates. And then with small molecule compounds, these are titrations plated left to right. And then another set of compounds, you could see compound-induced stabilization of the target. And these can generate nice dose response curves in which you can rank order compounds. And we found that for targets that are amenable to CETSA, that rank ordering is reproducible across experiments on different days. And so we've done this type of work for a number of targets, and we focused initially on targets that have other cell-based assays so we can benchmark the CETSA versus other cell-based readouts of activity. So this is data for LDHA again, where this is lactate production in cells, and this is the biochemical assay. And so what you'll notice is there's a number of compounds that do okay in the biochemical assay, but lose activity completely in the cell-based assay. Moving to the middle is the same. There's a number of compounds, again, that do okay in the biochemical assay, but are inactive in CETSA. But when you compare the CETSA and the other cell-based assay, there's nicer agreement between the two assays. And similar seen for ALDH1A1. This is an aldehyde dehydrogenase. And again, a number of compounds, and some that are quite good in the biochemical assay, but completely inactive in the cell-based assay or in CETSA. And if we compare another cell-based assay that measures activity of ALDH1A1 versus CETSA, again, you see a better correlation between these two experiments. And so we think that for targets, particularly if there's not another cell-based functional readout available, that we think that CETSA is a good approach to be able to rank order compounds and identify compounds that have good cell-based activity. Okay, so I'll touch on a couple of considerations for interpreting CETSA results. And this first one is related to membrane integrity. And so for the different cell lines that we've looked at, we've seen that membranes are disrupted around 60 C. And so for CETSA experiments where your aggregation temperature exceeds 60 C, this complicates interpretation in terms of whether the molecule can cross the membrane. And these CETSA experiments behave more like lysate experiments in some ways. And I'll touch on lysates versus intact cells a little bit more in the next slide. And the other consideration is that CETSA is prone to false negatives. And so we've seen many examples where we have evidence of small molecule binding to a target, including binding to the target in a cell-based environment, and yet there's no response in a CETSA experiment. And I think the most straightforward way to think about this is just that the most thermally unstable portion of a protein is not altered by ligand binding. And so particularly for big multi-domain proteins, if your small molecule binding event doesn't change the thermal stability of the weakest part of the protein, then you're not going to see a shift in the overall thermal stability of that protein. And so from a screening perspective, this is certainly something to consider, that that there may be a lot of binders that don't elicit a change in thermal stability in a CETSA assay. And to demonstrate this point, this is a kinase that we're working on, c-Abl. This is a well-known kinase that is targeted for a number of leukemias. And what you could see is that if we do a CETSA experiment with this molecule imatinib or Gleevec, which is a clinically used drug for leukemia that targets BCR-ABL, we get no thermal stabilization of the target with increasing compound. However, if we take the kinase-only domain of this protein and do this as a split Luke CETSA experiment, we do see thermal stabilization. And this holds true for other ABL molecules that are of different chemotypes that also we see no effect on the full-length protein, but we do see an effect on the kinase-only domain. And so I think the question that remains is that when we express this domain in cells, is it behaving normally per se? Is it interacting with the right partners? And are we accurately reflecting target engagement using that truncated protein? And so coming back to the lysates versus intact cells, this is a decision that's made typically early on in a CETSA experiment, or if you're going to be doing screening, whether to use lysates or intact cells. Of course, if you use intact cells, you maintain the ability to ask whether the molecule can get across the plasma membrane for intracellular targets. There's other considerations about protein-protein interactions and maintaining compartmentalization that may or may not be important for your particular target. I wanted to show this because this was something that surprised us a bit when we first did these experiments. And it's that the metabolite or ion concentrations in the cell may also really alter target engagement. And so in this situation, this is data for LDHA again. And in intact cells, you can see there's a bunch of compounds. Again, these are titrations left to right that show nice target engagement and stabilize the LDHA protein. But when you move to lysates, a lot of these engagement events either disappear or are greatly diminished. With the exception of this one compound here, which belongs to a different chemical series. And the only difference about this compound is this is the one that doesn't require cofactor for binding, whereas the rest of the molecules on this plate are known to require cofactor. And so it seems that when the cells are lysed, that the cofactor concentration drops precipitously, and then you no longer can see the binding event. And we've seen something very similar for ALDH1A1, where we get nice target engagement in cells. These three lines here are showing that we lose target engagement in lysates, but we can recover the binding event in a CETSA response by supplementing back its cofactor to approximate cellular concentrations. And so this is a consideration that when you're selecting cells or lysates, you may bias your hits based on a lot of different factors that go into whether or not the molecule binds. And so I'll expand just a little bit on what's in a melt profile. I think that in all of these CETSA experiments that are run, we see a single melt curve. But in reality, what underlies that melt curve is probably an average of a lot of different populations of a target in a cell, at least for some targets where there's different populations that may have different protein-protein interactions or different post-translational modifications, different localization. And so what we're really seeing is kind of the composite melting profile of all of those. And I think that's particularly important if you are looking for molecules, or if you think your molecules only interact with a subpopulation of that protein. As that subpopulation becomes smaller and smaller, it becomes more and more difficult to detect the engagement event by CETSA. And this goes further to that these different subpopulations and states may be different in different cell types. And so for some targets, we've seen very similar data across cell types for target engagement, but we also work on some targets where we see very different responses in terms of engagement across different cell types, and whether it's in normal cells versus disease cells, for instance. And so these are important considerations when thinking about doing a CETSA screen is selecting the cell type that's going to give you the types of compounds that are the most relevant. Okay, so I'll wrap up with two more quick pieces of data, and I wanted to just touch quickly on CETSA for screening. And I think that there's still a limited number of publications that have looked at how useful CETSA will be from a screening perspective for diverse collections of small molecules. But you could imagine why this would be really useful, because you could unlock potentially different binding sites on targets that may be distinct from, say, an active site that has homology across a family of proteins. You could maybe find more specific binding sites that then, for instance, you could couple with targeted degradation in PROTACs or other reasons that these binders at these unique sites might be useful. And so we've done some screening with lactate dehydrogenase again, and so I'll show some of the data there. This is a screen of 2,000 compounds from an oncology-focused library, and this is looking at lactate production. So again, the functional readout of what LDHA inhibition would do. And there's all kinds of hits in this library, and we think that a majority of them are off target. If you screen the same collection against the purified enzyme, there's very few hits. And this is a target that's had a historically low hit rate. But if we do the CETSA experiment, again, we get very few hits. There were 3 out of the primary screen, only 1 of which confirmed, and this is the known LDHA inhibitor. So at least in this case, the CETSA assay identified the relevant molecule. It didn't seem to be prone to false positives, but on the flip side, it also didn't find any molecules that potentially may bind outside of the active site. Of course, it's only 2,000 compounds, so it's a pretty limited set, but I think we could take home that it doesn't seem to be prone to false positives in this case. And the screen that we ran for this target was pretty reproducible across 15 plates. We saw a nice correlation of the potency for the positive control molecule. And so the last slide I'll show is this one, and we've been thinking a bit about whether or not we can use CETSA as a screening platform for rare diseases. We work on a number of different rare diseases, and the idea here would be to take a monogenic disease that is caused by a missense variant, something that would be destabilized and likely targeted to the proteasome for degradation, and then whether we can use CETSA as a platform to identify, for instance, small molecule chaperones. And so this is one rare disease protein we're working on, and 2 variants, missense variants that are thermally destabilized. And here's some example data of the approved drugs collection screened against one of these variants and looking for molecules that stabilize. And so we're interested in seeing whether this provides tractable hits, and then whether we can expand it out and look at additional rare disease proteins. Okay, so wrap up here, and hopefully I've conveyed that CETSA is valuable to demonstrate target engagement and useful for lead optimization and SAR, but there's also a number of limitations that are important to consider when implementing CETSA. And then I think the utility as an HTS platform is still something that remains to be determined, and I'd imagine over the next few years that there'll be additional data sets that are published by a number of groups that will really give us a better sense of whether or not it'll be a useful primary screening platform. And I will acknowledge my colleagues at NCATS. There was a lot of different folks that contributed to the work I presented today: biologists, chemists, engineers, compound management. So a big team effort to get this CETSA work completed that I showed. Okay, so I'll wrap up there and hand over to Lorena, and I look forward to taking questions at the end. Thank you.
Thank you, Mark. I'm going to try to take control so that I'm able to use my pointer. So let me just make sure that you can see my slides. Is that showing up?
Yep.
All right, great. Thanks very much. To presenter mode, to find my pointer. All right, so thank you very much, Mark. You know, as having introduced it, I appreciate that because you've covered a lot of the background. And as you already mentioned, we have been doing some very similar experiments and similar approaches. So as in the intro, I'm at GSK in Collegeville, Pennsylvania, and I'm in a high throughput screening department, but we also do mechanistic biology experiments and lead op support for programs. So how have we applied this? Again, I'm going to mention the detection format, which is similar but slightly different than what Mark described, and talk a little bit about full melt curves and then the isothermal version of the assay, whole cell and permeabilized cell versions, and then some of the optimization parameters that are really important, such as sensitivity to the temperature and the time for the melt, and then how we adapted the assay to 1536 well screening. I'm going to comment on destabilization at the end as well. All right, so as Mark mentioned, he was using the high-bit nanoluciferase split reporter. We are also using a split reporter approach, but our reporter is the beta-gal enzyme instead of the nanoluciferase. But again, it's an enzyme fragment complementation system where you tag with one small piece of the reporter and then you add the complementing fragment at detection. Just wanted to mention again, just to refresh your memory on the two different ways that we run the assays. So you can run a full temperature gradient and generate your melt curve and get your TM. And again, with a stabilizing compound, you might see that curve shift to the right. But you can also select a melt temperature and run at a single temperature. And that's how we would generally do our screening plate. So the entire plate gets treated at a single temperature, and then you might have your low signal here with a melted, and then your high can either be unmelted, added after the heat step, or a compound if you have that. So Mark had talked about whole cell and permeabilized cell assays, and I think this has been very useful for us. So just to go through the protocol, for the whole cell, you would just take your cells and incubate them with compounds for an hour or so, and then you would heat and then do your detection step. For the permeabilized cell system, we've been using a freeze thaw to permeabilize and kill the cells. Then you would just take those samples and then add your compound and incubate and do the heat and detection. And what we've been able to see in some cases is that you might have compounds that are inactive in the whole cell. So these are different compounds against one target. And for these compounds, they were inactive in the whole cell, but you can see that they engaged and stabilized when the cells were permeabilized. So in some cases, we do actually interpret these results as meaning that potentially the compounds have not entered the cells. And when we've done follow-up experiments to compare biochemical and cellular assay potencies, the potencies correlate in terms of biochemical, but not cell. So this would be an example where what we saw when we did a biochemical assay, in this case was an HTRF biochemical assay, pivoted against the CETSA results, we saw much improved correlation in the permeabilized cellular thermal shift. So in some cases, we do interpret this as that the compounds weren't getting into the cells, but engage the target when they're permeabilized. Again, I want to make a point about potency. And I've pulled this example from the literature. We have observed similar things from our work, and just haven't gotten the approval to present that externally. But this example shows what we've observed for multiple targets, which is that as you do your compound dose response curve at different temperatures, so in this example from left to right, as you increase your temperature that you're doing your target engagement experiment at, you can see there's a big shift in potency. So often you lose sensitivity as you increase the temperature of your experiment. In addition to that, we've also observed that as you increase the time that you do your heat treatment at, you can also lose a lot of potency. So I think, as we were optimizing assays in the beginning, we had to learn to resist the temptation to optimize the assay solely based on Z prime statistics and signal over background. Because as you can imagine, as you increase your temperature, you might get a much nicer signal over background for high control, which is unheated, all the way to heated. But as I've just shown you, you can lose sensitivity and lose the ability to detect your target engagement. And so the idea is that you really must balance your statistics with your sensitivity. And so that, again, can be challenging for certain targets, especially if you don't have good tool molecules to really understand what those parameters are. Also, as Mark mentioned, we have observed big differences in terms of both protein size and also subdomains of proteins behaving very differently from one another. So for example, you might see a full-length protein with a shallower melt curve and a smaller signal over background, and then take a truncate of that protein and see a much nicer melt curve. So we've observed that for multiple targets. This is also, in the literature, in a thermal proteome profiling experiment, you can actually see this. They had observed a general inverse correlation between protein size and the melt temperature, just showing that smaller proteins can be more stable. There is a limit to that, of course. As you get very, very small, they tend to be very unstable as well. So it is to your advantage to think about proteins, their size, and their domains, especially if you're going to do an expression experiment. You might want to entertain the idea of making several different constructs to test. So Mark also talked about using a PCR thermocycler for melt curves, and we still use this in our medium throughput and our mechanistic experiments. These detailed types of melt curves allow you to titrate your target expression, to generate full melt curves under different conditions, and really optimize your assay and get detailed experiments. But for considering higher throughput, so our full diversity collection can be between 1 and 2 million compounds. If you're using a PCR cycler to heat your plates, it can take up to 5 to 10 minutes per plate to heat your plate. Now, Mark had shown the heat block solution, and of course you could have a bunch of heat blocks or a bunch of PCR cyclers to increase your throughput. But just consider the heat time for 2 million compounds, for example. It would take hours and hours of time just to do the heat step. And the PCR plates were really not very compatible with our liquid handling and automation. They can warp, so they're really not good for automation. So we had to come up with some different solutions for the heating and the plates. So what we did was to develop a water bath heating method. And so in this case, we have to seal the plates. And then what we do is we put them into a rack, and then we use a nice low-tech bungee cord to strap the plates into the rack. And then we turn the whole rack sideways and put it into a heated water bath. And then at the end, you just take the plates out, you clean off the water, pull the seal off, and then add your detection reagent. So again, it's a nice, inexpensive solution for heating. Obviously, you can only use it for the isothermal, but we did have to optimize the plates and the plate sealing. And so again, plates that have, for example, a ridge around the edge, this ridge can trap a lot of water. And so when you pull your seal off, it drags all the water into your wells, and it confuses your assay results. And so again, multiple plate types were tested, and there are several different plate types that work well. And again, for the plate sealer, you really need to have a seal that can withstand that water and the heat. And so this is one of the ones that we use. But in the end, when you use these methods, you can generate very nice assay statistics, very consistent results, and develop assays that are fit for screening. Just want to show an example of, for example, a batch of screening data, 30,000 compounds. And what you can see, again, is just really excellent screening statistics, low hit rates. So for example, at 3 standard deviations, that's actually quite low. We might go all the way to 5 or 6 standard deviations. But for normal screening statistics, we would end up, if we increase the standard deviation, you'd get up to maybe or down to below a 1% hit rate. So really nice low hit rates is expected for this assay that we also use in hit qualification. But what about the destabilizers? So as you noticed on the last slide, I had shown that we were looking for stabilizers in this case, but what I hope you can see is that there are a lot more hits down here in the destabilization. And so what we have observed generally is that there is a much higher nuisance and interference rate in the destabilization direction for this assay. But when we have gone through and done specificity testing and triaged it, we can see that some of these are real and target specific. So there's just a lot more nuisance in there, but there are also some real hits in that space. And so I just wanted to mention this as a sort of a new trend and direction. It's becoming more and more evident that a lot of compounds that do engage targets actually do cause destabilization. I pulled something from the literature here. In this case, again, it was a thermal proteome experiment where thousands of proteins were looked at in an extract using mass spec readout. And, in the paper, you can see that they've pulled out multiple examples where known compounds against known targets destabilized them or stabilized them. And there were multiple examples there. And so what we have observed in some cases that for some targets, most compounds, known compounds, will destabilize instead of stabilize. So I think this is a real emerging trend and something to watch in the CETSA space. So I'll just close out with some challenges, again, to reiterate some of the things that Mark said. Not all proteins are amenable to thermal shift. Some of them don't melt well. Some of them can't be stabilized well with small molecules. For the high throughput approach, you do need to engineer in the tag and express your tag target protein. And then there's that false negative problem, which is that not all compounds that bind to targets will produce a thermal stabilization. So again, if you get a negative result, it doesn't mean that the compound does not bind. But it has been a very useful assay. And so I just wanted to go through some of the areas throughout the early drug discovery process that we've applied CETSA and where it's been useful. First, in hit qualification, in following up from a biochemical, a cellular, or even phenotypic high throughput screen, we've been able to take confirmation lists or hit lists and run them through and really identify a prioritized list of compounds that truly look like they bind the target. And at this stage, we can also run a specificity assay to look at which ones bind specifically. We have found this to be especially useful when the protein can't be purified or when there seem to be challenges with biophysics. So if for whatever reason you're trying to confirm hits with SPR or other biophysical methods and you're having challenges, CETSA has been really useful in that space. Again, Mark showed the lead optimization example. We have used it in that space as well. You can look at rank ordering of compounds and different series of compounds. And again, we've seen some really interesting results where some series might stabilize and some series might destabilize the target. So very useful there. We are exploring the hit ID space, as I showed, for targets where you might be interested in a PROTAC approach. So you don't need functional activity. You're just looking for a binder that you can build into a PROTAC molecule. And you've seen our 1536 well screen adaptation that works very, very well. I think other uses that we've seen, and it'd be very useful, for example, when you're early in a target and you're trying to understand a tool molecule that might have been described in the literature, for example, it's been very useful to show that that target and that molecule engages that target, to confirm the literature. So it actually increases what we see as a tractability score on our targets if we can show that you have small molecule engagement. Again, you can inform on cell versus biochem assay with the caveats that Mark talked about. I completely agree. We have seen some unusual behaviors in comparing lysate or whole cell, either by dilution of cofactors or differences in things that happen when you create a lysate. But in some cases, it's been quite useful to compare the whole cell and lysate versions of an assay. The other area that's been great is to use target specificity results there. So you can build an assay for an unrelated or a related target or an animal ortholog, for example. So if you know your in vivo model before clinical is going to be a rat or a mouse model, you can take your animal ortholog and check your compounds to make sure that they're engaging the animal ortholog as well before you go into your animal studies. So very useful in multiple spaces along the drug discovery path. Much of the work I showed you has been done by several of these individuals, Anna and Bobby, and then others have contributed, Ashley, Maggie, and Josh, my line leadership, and then some of the early work at GSK was done by Mark Holbert and others. I also would like to thank all the sample management and the reagent generation groups at GSK for providing reagents and compounds. So with that, thank you very much. And we'd like to go into questions. I would just like to remind you that we are using this chat function. So you can go into the chat and select to all panelists, and Sharon will read the questions. So Mark, both of us, you can, come off mute video, and they can be for either one of us. So thanks very much.
Yeah, thank you, Lorena and Mark. Great presentation. I'm very excited about what I learned today. So there are a couple of questions right now. So the first one is, are certain types of targets more amenable than others?
Mark, do you want to take a stab?
Yeah, sure. Yes, I think definitely. I think that there's particular types of targets that are probably less likely to work, like things with lots of multi-pass transmembrane domains. Those seem to be quite thermally unstable and maybe less likely that you'll get a small molecule binding event that will change the overall stability. I think there's quite a bit of data in the literature for particular targets where there's a lot more CETSA data, kinases, and so it seems like there's particular targets that are well suited. And so yeah, I think that there's going to be some that are better. And if your protein is related to a target that already has CETSA data on it, then I think it's more likely to work. It gets a little more challenging when you're moving into a new space. And particularly if that new space doesn't include good tool molecules, then it's really hard to actually gauge whether or not CETSA is ever going to work. You could kind of go in circles for a while, but I think for some targets, there's quite a bit of literature data out there that you can rely on.
Yeah, I agree. And any biophysical precedence, for example, is a good thing. I'm going to follow up a little bit more on the membrane part. So there are publications showing that you can increase your target range by adding some, detergents and things to your cell lysates to access some of the membrane proteins. But I do believe, from what I've seen, that those require very individual optimization. So there's not one set protocol that you can use, for example, for all membrane proteins. It would take some real good investigation and optimization to get at things like membrane targets. That said, we have worked on some intracellular membrane associated targets, and those seem to behave quite well, even for some that are not that amenable to protein purification. So it does expand the space, I think, pretty well in some cases.
Excellent, thank you. So next question, can you comment on advantages and disadvantages of the HiBET versus DiscoverX platforms? And I'm hoping I'm pronouncing right, HiBET.
Yes.
Okay, good.
Wow, that's a really good question. We've done minimal comparison. So as I mentioned, most of the work that we've done has been with the Eurofins DiscoverX beta-galactosidase EPL tag. So I don't really have a lot of comparison data. And maybe Mark, you can comment on that.
Yeah, no, we also haven't done any direct comparisons. I mean, I think conceptually they're very similar. I mean, the size of the tag is different, the detection modality is different. You'll probably get different artifacts in terms of molecules, especially for these destabilizer type things where you have more artifacts to sift through. I think those would be different, but I think they're pretty similar in concept.
Yeah, I will make the comment though that and you showed the example of the comparison of the Western blot to the reporter version. And in cases where we have looked and done that experiment, I think we do see in general very nice correlation between a naked protein Western blot approach and the reporter approach. And I know you have those examples as well, Mark. So overall, the small tags do not seem to affect the protein melt very much. And that's one of the advantages of these enzyme fragment complementation approaches is that the tags are quite small. You know, the high bit is 11 amino acids and ours is 40. The EPL tag's about 40, 43, 42 to 44 amino acids. So while it's a little bit bigger, again, it doesn't seem to impact the melt of the protein that you're tagging.
Good, excellent. So next question is, are PPI more challenging than enzymes?
Well, protein-protein interactions. So people have done some off-target effect experiments using, for example, the thermal proteome experiments. But most of for example, if you have a signaling molecule that binds to one target, but then induces phosphorylation or modifications or changes in the protein-protein interactions downstream, you can detect some of the changes in the pathway targets as well. I don't know of an example where this technology is very confirmed or useful in actually detecting the protein-protein interaction itself. I think you would have to empirically test it. There are certainly examples of things like peptide target interactions that you can look at, but we have not used it for a protein-protein interaction experiment per se. So I don't know, Mark, if you've done that or not.
No, we haven't. We also haven't done that. But I think it seems like it could be done. I think it also kind of gets to the classic interpretation of a CETSA shift is that your molecule's directly engaging, right? And it's clearly the case for a lot of molecules and proteins, but you could imagine, and I think this is the protein-protein interaction idea, is if the molecule broke that interaction, you might get a thermal shift in your protein that would occur really quickly, right? And it would look identical to a direct engagement of that target versus binding to a partner and falling apart. So I think there's room to use CETSA in that format, and it seems like that's that it would be expected to occur and may actually explain some CETSA results, but I don't know how much of that has been demonstrated for changes in thermal stability as a protein-protein interaction.
Yeah, I agree completely. There could be space for it. I think from the thermal proteome profiling experiments, like I said, if you dig through some of those mass spec proteome experiments, people have observed changes in pathway. I just wonder actually if we could find an example of something that was just a protein-protein interaction. I don't know for sure, but I agree with you, Mark. I think it's completely feasible and could very well work and be useful in that space.
All right, so that's the questions I have. For now, I have a question just related to the water bath, Lorena. So how long did it take that implementation since it was something new?
The assay development, we looked at a few different ways of doing it. But by the time we decided to try the water bath and implement, to fully optimize for targets, it took months, but I think some of the initial experiments, were done within a matter of weeks to show the proof of concept. So weeks to months initially. Now it's very established. So we take those plates right in, test melt curves. You can do, those experiments right off the bat now.
Okay, excellent. One more, Kim. Had you considered any fully automated methods for the heating step?
I don't know about you, Mark. We do put them in stacks, and I'll just make the comment that in general nowadays, even for high throughput screens in other formats, we use a batch-based approach for some screens anyway. Some of them are more automated than others, but we often just have people stacking plates, and it can go quite quickly that way. So we don't always use automated platforms and robots to do our plate batches for HTSs anyway. So I think it's pretty comparable process to high throughput screening for many of our targets and assays.
Okay, excellent.
We've thought a little bit about automating, like having a hotel of heat blocks where you could kind of heat them in sequence. I think one of the big engineering challenges is getting top pressure on all of those plates. So I think we don't have a solution for it yet. It's something that we were interested in trying to do, but have not actually fully automated it. It's still done by hand on the heat block. I think the water bath idea, though, it makes a lot of sense for high throughput. And I was going to ask Lorena, can you float plates or do you have to submerge them?
Well, you know what? If you float them on top, it limits your space as well. And then we
Mhm.
just found it didn't heat as well. I think it's similar to your idea where you clamp the top down and heat from both sides. And I
Mhm.
think that's really helpful to get quicker, more even heating. So we just found it worked much better to completely submerge the plate and get really even heating and results that way. So it's interesting adaptations, both cases, but I think the end result is the same.
We
Yep.
have one of our participants that they
Well,
say,
we're very short on time, but let's take a quick one and then wrap up.
yeah, they say that just to consider looking into Ineco ovens, and they can also apply pressure to the top.
Uh-huh.
So
Ovens, yeah. I think the idea behind the air we thought wasn't as good as a heat conductor, so we were concerned about air, and I think that's the same reason, Mark, you went to the metal. Water and metal are better conductors than air. But, we're always happy to entertain new ideas, but the flip side is what we're doing right now works very, very well and is very high throughput also. All right, we should wrap up, so I'm going to hand it over to Rodney, I think, to wrap up. So let me just go to the next slide.
Yes, thank you. Thank you, Lorena and Mark, for excellent presentation, and Sharon for handling the questions and introduction. There will be a recording of this available, and I will send it out to the people who registered for this. We had a very high attendance today. We had 136 people interested in attending, and we want to thank you for your interest in these events. Please provide proposals for future scientific presentations. And if you'd like to organize or if you'd like to present, just write to talks.phl@lrig.org with your proposal. We will have our next event. We'll have two speakers on the 5th of May, the first Wednesday in May. So there will be information coming out. LRIG.org will give you information and we will distribute it in the usual channels. So again, thank you very much for your attendance and have a nice day. Signing off.